Hey everyone, welcome to this explainer. You know what? We're actually going to skip the usual agenda today because, well, we're packing our bags for a totally different kind of trip. We're going on a journey inward. We're going to literally leave the visible, everyday world behind us and dive headfirst into the microscopic universe that's secretly running the whole show. We are talking about the fundamental architecture of absolutely everything that lives and breathes around you. Now, to really understand life, we've got to go all the way back to the very beginning. I'm talking about some of the most extreme environments you can possibly imagine. Picture this, the hot springs of Puga Valley over in Ladakh, India. It's a freezing climate, right? But these waters are sitting right near the boiling point. And scientists actually believe these intense, crazy environments closely mirror what the early Earth looked like about 3.5 billion years ago. So what did researchers find around these springs? Well, they found these rapid formations of calcium carbonate deposits. And it's highly likely that these rocky deposits actually protected the very first fragile organic molecules from harmful radiation and extreme weather. They basically helped form the very first protective boundaries. And those boundaries, yeah, they are exactly what allowed the first heat-loving bacteria to even exist in the first place. So that primitive protective boundary, that was literally the birth of the cell as we know it, the fundamental unit of structure and function. And honestly, whether you're a microscopic, heat-loving bacterium just hanging out in a boiling spring, or a complex human walking around with trillions of these units working together, this microscopic boundary is the ultimate theater where life actually happens. Even when these units group up into vastly complex tissues or organs or whole organ systems, the cell itself, it remains the absolute core building block of the whole shebang. But you might be wondering, if they're so important, why did humanity go through almost all of its history without even knowing these fundamental units existed? Well, it all comes down to our own biological limits. Try this sometime. Take a piece of paper, draw two tiny dots, and slowly move them closer and closer together. Eventually, you'll hit a physical threshold where your eye just can't tell them apart anymore. From a normal reading distance, if 2 points are separated by less than 0.1 millimeters, they just blur into a single solitary point. That 0.1 millimeter mark is the absolute limit of resolution for the naked human eye. And because almost all cells are way smaller than this limit, they were completely invisible to us for thousands of years. Okay, let's dive into this. To bypass our own biological blind spots, we had to get a little creative. On the left here, we have Robert Hooke's ingenious solution from way back in 1665. He built this light microscope with about 200 to 300 times magnification. He looked at a super thin slice of cork, saw these tiny little box-like compartments, and famously decided to name them cells. And hey, we still use modern versions of light microscopes today, using combinations of convex lenses to magnify visible light. But if we really want to see the true, fine details of a cell structure, we need the incredible piece of machinery on the right, the electron microscope. Instead of bouncing light, it uses a literal beam of electrons. This lets us see structures all the way down to the nanometer scale, which, by the way, is 1/1000th of a meter. The clarity is just absolutely mind-blowing. So, once we finally have the tech to actually see a cell, how on earth do we figure out its actual size? It's actually a brilliant yet surprisingly simple bit of math. First, you just place a transparent ruler right on the microscope stage and measure the diameter of your circular field of view in millimeters. Step 2, you convert that measurement into micrometers by multiplying it by 1,000, so a 5 millimeter field becomes 5,000 micrometers. Easy, right? Step 3, you swap out the ruler for your actual slide, let's say it's an onion peel, and you literally just count exactly how many cells fit in a straight line straight across that diameter. And finally, step 4, you divide the field size by that cell count. If your field is 5,000 micrometers wide and you count exactly 25 cells across it, boom, your single onion cell is 200 micrometers wide. It's such a simple way to measure the invisible. All right, so we know they are there and we know exactly how big they are, but a cell isn't just some isolated little box cut off from the world. For any living thing to survive, its cells have to constantly interact with one another and with their outside environment. So how do these tiny compartments actually pull that off? How do they pull in the good nutrients, expel the toxic waste, and communicate with the rest of the body? Well, the secret lies entirely in that original protective boundary we talked about earlier. That incredibly thin outer covering is what completely defines the cell's individuality. And that boundary is known by a pretty famous name in biology, the fluid mosaic model. Get this, it is only 7 to 10 nanometers thick. It's built primarily from what's called a lipid bilayer. But here's the kicker. It's not a rigid, solid wall. It's highly fluid. The molecules making it up are constantly shifting around, flipping, and rotating. Meanwhile, you have these embedded proteins that act like highly trained gatekeepers, deciding exactly what gets the VIP pass to come inside and what gets kicked to the curb. So rather than a static, boring shell, you've got to think of it as a bustling, dynamic mosaic, where every single piece is constantly shifting to keep the internal environment absolutely perfect. Now, what's really interesting about this slide is that we can actually see these selective gatekeepers in action using a super simple kitchen experiment. Seriously, try this at home. Take a potato, cut it into 2 roughly equal pieces, and weigh them so you have a baseline. Then drop one piece into a beaker, let's call it beaker A, which just has plain tap water. Then take the second piece and drop it into beaker B, which is filled with a highly concentrated 20% salt or sugar solution. And then you just leave them alone for about an hour. What we're doing here is effectively forcing the cells of the potato to deal with 2 wildly different external environments. To really grasp what happens in those beakers, we need to talk about osmosis. Osmosis is basically how water chases solute concentrations across a membrane. Think of it like this. If the environment outside the cell matches the inside perfectly, it's called isotonic, and everything is perfectly balanced. No stress. But if the outside environment has fewer solutes than the inside, it's hypotonic. And if it's packed with them, like our heavy salt solution, it's hypertonic. Because those protein gatekeepers we mentioned are super picky about what solutes can pass through, the cell membrane is constantly trying to balance the scales by moving water instead. Okay, let's look at the actual results of our potato experiment. In beaker A, that plain water is a hypotonic solution. Because the potato's internal solute concentration is way higher than the plain water surrounding it, water aggressively rushes into the potato cells through the membrane to try and balance things out. The result? The potato piece physically swells up and actually gains weight. But over in beaker B, that 20% salt solution is fiercely hypertonic. The concentration of salt outside is much, much higher than inside the potato. So the water is rapidly drawn out of the potato cells, leaving the potato piece looking visibly shrunken and causing it to lose weight. And we can actually see this hypertonic shrinking happen in real time under a microscope, which is just so cool. Take a look at these real leaf plant cells. On the left, you see them in their normal, healthy state. But when we add a 20% sugar solution, notice what happens on the right. After just half an hour, the rigid outer wall is still holding its ground, but the inner contents of the cell, they've completely collapsed in on themselves. The space between that delicate, fluid cell membrane and the rigid outer wall has drastically expanded as all the water just completely drained out. And this brilliantly illustrates a really crucial difference between plant and animal biology. You see, plants, fungi, and bacteria feature this tough extra layer outside their cell membrane called the cell wall. Think about it. A plant can exactly uproot itself and run away from a severe drought, a heavy rainstorm, or harsh winds. So it relies entirely on this rigid structure to stay upright, even when it's rapidly losing water. Animal cells like ours, on the other hand, we don't have or need this armor. We only have that thin, fluid mosaic cell membrane, which makes our cells highly susceptible to shrinking or even bursting if their environment changes drastically. This structural divide is stunningly obvious when you put them side by side under the microscope. Notice how the onion cells on the left are highly organized, locked neatly into place like bricks in a wall, all thanks to that rigid structure. Now, in stark contrast, look at the human cheek cells on the right. Because they lack that cell wall, they are loose, irregularly shaped, and honestly, a little blob-like. But here's the amazing part. Both types contain all the incredibly complex instructions for life, and both use those dynamic, fluid mosaic membranes, yet their outer boundaries dictate entirely different physical structures. So look how far we've come. We've traveled from the boiling, harsh extremes of the early Earth, looked through the incredible nanometer lens of the electron microscope, and zoomed right down to the fluid gates that protect every single cell. The next time you look out the window at a towering tree, or pet your dog, or even just look down at your own hand, I want you to consider what's happening just beyond that 0.1 millimeter limit of your vision. Trillions of microscopic universes are constantly communicating, filtering, and fighting to keep you alive right this very second. It kind of makes you wonder, doesn't it? Who was really in charge here? You or the trillions of invisible cells running the show right under your nose? Thanks so much for joining me on this journey inward, and I'll catch you on the next explainer.